Good morning, everybody. How's everybody doing? Awesome. It's really, really a tremendous pleasure to be here. You know, I have the privilege of being, if I walk out here, can you hear me? All right, so I have the privilege of being able to give talks, you know, in lots of different places and being able to speak to students, particularly Latino students and people just starting out in their career is one of the things I enjoy the most. So it really is a heartfelt thank you for the invitation, and I want to really be here to tell you a little bit about the science that we're doing, but also during the course of today, if people have questions about my career trajectory, happy to answer those and tell you how I got to where I am, which is, in many ways, I've always felt I sort of Forrest Gumped my way through life and got very lucky to be where I am, and so happy to tell you all those stories. I want to begin by, on the science side, telling you a little bit about the work we do in my lab. I started life out wanting to be a population geneticist, which is kind of odd. A lot of people don't do that when they're 19. And then I got, you know, very interested in all the fields that are related to it, and so I studied both math and biology when I was an undergraduate, and that really sort of has led me to set up the program that we've got now. So our goal is to understand the genetic basis of traits of human importance. So that could be traits in humans, so disease susceptibility, for example, or adaptation. Traits in human pathogens, so we do study a lot of the bugs that infect us. Our pets, I've always been very interested in dogs, particularly dogs as a model system for rapid evolutionary change, as well as the foods we eat, our closest relative to the primates, and so on. And ultimately, our goal is to go out and answer important questions in biology, anthropology, and medicine. And several years ago, when I was at Cornell, I used to be at Cornell in the Department of Statistics, I decided that I wanted to expand my research program. So after I got tenure doing a lot of very sort of rigorous mathy things, I became passionate about underrepresentation in genomics, and in particular, the fact that most genetic studies have only been done in populations of European descent. And that really sort of kind of grew a passion in me to sort of tackle the things that I'm now tackling. And in order to do that, I had to kind of vertically integrate my research program. And so today, we actually go from people doing sampling in the field, both for humans and for example, dogs, to the development of new sequencing technologies, to the development of functional assays in the lab to test our hypotheses, to the development of new theory and methods that then inform the set of experiments that we want to do. And so in my lab, I've got people that run the gamut from applied mathematicians and physicists through geneticists, through anthropologists, through ethicists, because there's a ton of our work that impinges on lots of different areas. And again, sort of happy to tell you all about that. In particular, you know, most of what I'll be talking about today has to do with DNA variation. And a question you might be asking yourself is, why the hell do we want to study DNA variation? Why is it important? Why is it interesting? I would say from an aesthetic point of view, because all the questions we want to answer are ultimately aesthetic, right? They're the things that we think are beautiful and interesting. One question that's always fascinated me is, why are we so phenotypically diverse, right? If we think of humans as a species, we vary so much in body size, in height, in weight, in skin pigmentation. It's just beautiful, right? And we don't really understand all the genes that underlie that. And so, for example, I'll tell you a story today about kids from the Solomon Islands that have this spectacularly beautiful blonde hair. And we set out to answer a very simple, like kindergarten level question, which is, do these people have blonde hair for the same reason that this little girl from, you know, we could say Sweden or something has blonde hair? And I'll tell you that story. The other reason we might want to understand genetic variation is because we're currently in a phase where we're rapidly getting DNA information on many, many, many people. So most of you will have the opportunity to have your genome sequenced. Whether or not you may want to do that is a question that you're going to need to grapple with, but that's coming. And Obama announced in his State of the Union address a personalized medicine initiative that goes out and he's going to try to recruit 1 million people. 1 million people isn't going to be enough, I can tell you that right now. We're probably going to end up, from a federal government point of view, genome sequencing maybe 10 million people. I think that's going to be a fundamental paradigm shift, so we're going to be seeing kind of a creative destruction of epidemiology. If you think about epidemiology as a field, it's been set up to follow a group of people by investigators, and that does not scale well. It's very expensive. What's going to end up happening is it's going to be a direct-to-consumer, patient, participant kind of thing. So we're going to be probably recruiting something like, I don't know, 10% of Facebook or 20% of Facebook, right? Now you're really thinking about the kind of large numbers, big data, and all the genetic information that goes along with that. One huge problem is that most of the databases that we have on genes and their relationship to traits only come from populations of European descent. And like the numbers, like 95% of the participants in these large-scale genetic studies are of European descent. I think that's a problem for all kinds of ethical and inclusionary reasons, but it's also a problem from a scientific point of view, and I want to kind of convince you of that. And then most of the talk is actually going to focus on what I find the coolest stuff we're doing, which has to do with ancient DNA and the opportunities there to study historical events, the great human diasporas that have not left written records, and I'll focus a little bit on those questions. Okay, so several years ago, we began a big project called the 1,000 Genomes Project. How many of you have heard of the 1,000 Genomes? Any of you? Okay, so the 1,000 Genomes is probably the largest international effort to document genetic variation. We set out to sequence 2,500 genomes, but that actually came slightly late in the game. Initially, it really was 1,000 genomes, and when the project was set up, the goal was to sequence 400 people from Europe, 400 people from Africa, and 400 people from China, or from East Asia. And we were at a meeting in Cold Spring Harbor, and I said, well, look, I don't want to be a stickler, but that's 1,200 people, number one. And then secondly, I said, how are we going to have a project called the 1,000 Genomes Project and not include people from the Americas, right? That's really, quite screwed up, right? Like, even just from a political point of view. And it got a very interesting set of discussions started. So some folks said, well, Native Americans don't want to participate in genetic studies. And I said, well, excuse me, have we asked them recently if they want to participate? And just because, and I said, I'll defend their right not to participate. They have every right, vulnerable populations have every right not to participate. That doesn't get you off the hook from including them. And I said, well, what if we, left them alone and invited them to participate, but what if we sampled, say, in, I don't know, Mexico City or New York or Miami? And then people sort of crinkled up their nose and they said, but they're admixed. And I said, okay, what the hell does that mean? What kind of Victorian notions of race do you have that you think those of us that have ancestry from different continents, we don't have pure genomes? Is that what you're trying to tell us? Can we go talk to the Congressional Black Caucus and the Congressional Hispanic Caucus? Because I think they'd be very interested in this point of view. And that led us to really sort of marshal the proper scientific arguments about why it was important to broaden representation. And so after we made our cogent scientific arguments, we got 500 samples from the Americas added to the project. And I didn't do this alone. In fact, I was part of a group. I just could be the spokesman for the group. And so we sequenced individuals, Mexicans from Los Angeles that had been collected as part of a previous project, African Americans from Oklahoma, and then we included and did new sampling in Puerto Rico, in Peru, in Colombia, and then our friends said, well, what about the Indian subcontinent? I said, yeah, what about the Indian subcontinent? That's 1 billion people. How do we have a project not include them? And so then we got these people included, which is great. And that today is, as I said, one of the biggest projects. And one of the things that we've learned as part of this work is that there's a tremendous amount of human genetic variation. And the structure of that variation is actually pretty particular. And one way of looking at this is, for those of you who are interested, all of the details are in this PNAS paper. This is a little kind of commentary that I wrote for Nature on this. And it turns out that when we think about common genetic variants, so common genetic variants would be DNA polymorphisms that were present at the time of the great human diasporas 50 to 100,000 years ago as the populations that gave rise to diversity outside of Africa, left Africa. Well, that variation, that which reaches 15, 20, 25% frequency in most human populations, is not surprisingly shared across populations. And so if we think about a pair of European populations, this is a measure of exchangeability. They're quite exchangeable. Even sort of a Chinese and European population or an African and a European population are going to share variation. Okay, so that's good and nice and really sort of recapitulates what you've always heard in biology, that most variations within and not among populations and so on. And that has to do with the fact that when we measure variation, we use heterozygosity, right? So it's 2x, 1 minus x, and that's of course going to be heavily weighted by the common variance. Now, most of this variation is actually pretty rare. Most DNA variation is not common. It actually follows a distribution that looks quite exponentially distributed. In fact, it's even more. It looks like kind of like a very skewed gamma distribution towards the rare alleles. Now, this rare variation, which we can pick an arbitrary threshold, let's say 1%, this variation tends to be extraordinarily population private. But when we're talking about 1% of humanity, right, and we're talking about, say, 12 billion chromosomes, right, that's still a lot of people, right? It's a lot of chromosomes. There's hundreds of millions of chromosomes that carry this variation. And if most of what we are thinking about now, and if you think about the latest set of genetic studies that are all looking to query rare variation, now this issue of which populations we study becomes fundamental, right? Because we're going to find common genetic variants in Europeans that will replicate, but there's going to be a ton of variation that's going to be private to populations that unless we study them, we're not going to find, okay? And that's really sort of a fundamental point that we try to make over and over and over again. And I want to give you a couple of examples of that. So what we did is really, over the last, 7, 8 years, have worked to broaden representation in medical genomics. So that project I told you about, the 1,000 Genomes Project, you see these little green Google Map dots. That's where those populations are. And you can see they are a smattering of populations all over the world. Before that, there was a big project called the Human Genome Diversity Panel, led out of Stanford by Luigi Luca Cavalli-Sforza, who's really sort of the grandfather of human population genetics. And his populations are in yellow here, and you can see that there's more there, and they cover more of the world, but there's still pockets that had never been studied. So in red are the places where we've made a lot of investments. So I'll tell you a story about populations from West Africa, where we went out and specifically sampled these populations because they're overrepresented in slave ship records, and one of the things we want to study is the Middle Passage. African Americans have lost a huge amount of their history, and using genetics we can kind of reclaim that history. We've studied different populations in South Africa, particularly because we believe anatomically modern humans originated in South Africa and then spread out all over the world. So that's a place that has a tremendous amount of human genetic variation. And we know next to nothing about the relationship between genes and phenotype. So one of the things we're studying is skin pigmentation in South Africa. And in South Africa, there are some indigenous populations that have skin as light as many of you in the room. Right? And you say, okay, well, is that ancestral? Is it derived? We don't even know what the ancestral skin pigmentation of humans was. Right? We know anatomically modern humans arose in Africa. That doesn't mean that they have the very dark skin pigmentation that we associate with West Africa. That's actually a quite highly derived trait. So these are kind of these fundamental questions that we don't know the answer to. We studied, most of our work is in Latin America, so we had a big project, for example, in Mexico, where we studied 25 different indigenous groups in Mexico and how they relate to other populations. We're studying altitude adaptation in Peru, and so on. And then I'm going to tell you a lot about this group in Europe that we studied, partly because that's where there's been a tremendous amount of resources and investment made. And I'm going to try to make the argument that most of what we can now do in Europe, we should be able to do in the rest of the world. Okay? The last bit I want to tell you about in terms of sort of medical relevance is just one example. And we have a big project called the Clinical Genome Project, where we're trying to aggregate all the clinical genetic testing data in the country and come up with estimates of the frequencies for these genes that are clinically very important. This is one such gene. It's called the HLA. It's the part of your genome that has to do with your immune response. And it turns out that at HLA, there are lots of different alleles, lots of different gene variants, and one of these is just called B star 5701. These things often have like serial number names. Okay, you're not going to be tested on this, but this allele, okay, happens to have a very interesting property, and that property is if you are HIV positive and you take a drug called abacavir, which is the #1 drug prescribed as part of the triple cocktail, and you happen to carry this allele, you will get toxically sick. The first time you get it, you'll have a terrible immune reaction because of this gene that you carry. And if you take it again, you die. Okay? So this is a very important clinical, gene variant that we should all know about, right? Because if you're going to get this therapy, you want to make sure not to get this particular drug. Well, here's the distribution of that mutation, right? And it turns out, and my dad's an HIV doctor, and I asked him, Dad, how often do you test your patients? He goes, oh no, I mostly see, African American and white patients, so we don't ever genotype them. And that sometimes makes sense because in West Africa, the frequency of this thing is really low. It's about 2 to 3% in Europeans, but if you happen to be Indian Asian, it's up at 20%, right? So this is super, super relevant to a subset of the population. The next highest population is the Maasai in Kenya, and there's no rhyme or reason to that. It just happens to be that's the way life is. And then a big population in Utah that has a founder allele and so on. And so, this is one of these things where we know this is clinically really important, and we should be testing for it and understanding the global distribution because it will really impact people's health and death, okay? We don't know most of this for most of the alleles, okay? And this is one of the things we're very passionate about. All right, so now on to some stories. The first story I want to tell you is about blonde hair in Melanesia. So several years ago, this guy, Sean Miles, came to my lab, and he had done his PhD in a set of populations known as the Solomon Islands. The Solomon Islands is one of the most remote places on Earth. It's in the middle of the Pacific Ocean. After you pass Papua New Guinea and you keep going east, right, like towards Easter Island. It's a population mostly of Melanesian descent, and they have this incredibly striking blonde hair phenotype. And Sean came to me and he said, Carlos, I want to study the genetic basis of blonde hair. And I said, Sean, look at this kid's US serviceman's jacket. I can tell you the genetic basis of blonde hair, right? Dad had blonde hair, he was European, it's sort of a Captain Cook allele that got introduced. And he said, no, I think it's going to be something different. I said, okay, look, if you can get some money and you can do the sampling, then we'll think about the project. Figuring that if any of you know postdocs, that they'll soon forget about it and move on to something else. But also, if you know postdocs, they tend to be really, really stubborn. And so Sean got the money and went out in the field. And here's Sean out in the Solomon Islands with his field assistant, who's another sort of PhD scientist. They're doing spectrophotometric measurements of hair. Okay? And this is where the Solomons are. As I said, here's Papua New Guinea. You're sort of, you're looking here towards the Pacific. We're somewhere kind of like over here where you can't even see the pointer. Okay? So then he came back to Stanford. He sampled 1,000 kids, and he said, Carlos, I want to now characterize them genetically. And I said, Sean, that's a huge amount of money. Each of these arrays costs like $250, right? We don't have $250,000 just sitting in the bank. And then Sean said, well, what if we just look at the extremes of the distribution? Let's look at the kids with the darkest hair and the blondest hair. And I said, yeah, that sounds kind of nuts. But you know what? Like, we'll burn 100 arrays on it. When this doesn't work, then you're going to have to go find another project. Okay? So we ran the arrays, and the way this works is that you have, in this particular case, about 1 million markers in the human genome. At each of those 1 million markers, you look for allelic differences, so differences in frequency among cases and controls. You then do a chi-square test, and the p-value distribution tells you where there's a strong association. So this is what one of these things looks like. Here is the human genome. This is chromosome 1, chromosome 2, and so on. Each of these is a marker. And this is what is sometimes called a Manhattan plot. It's supposed to remind you of the skyscrapers in Manhattan. This is the -10 log p-value. I like to call this our Dubai plot, right? There's, this one huge tower of association. For those of you who follow this, you know that one way of figuring out whether this is a real association or not is to do a QQ plot. And what I can tell you is that this would be the null distribution. The fact that we've got all these points and that they all map to one place tells us that this is likely to be a really big and true hit. This is your sort of Bonferroni correction for multiple testing. Okay? When we look inside this region, we got very, very lucky. And the reason we got lucky is that there's only one real gene in that region, and it's not some unknown gene. It turns out to be a gene that's been very well studied, a gene called tyrosinase-related protein 1. The reason this gene is interesting to us is that it is a gene that is expressed in melanocytes. Melanocytes are the cells that produce melanin, which gives you skin pigmentation. At this point, as a PI, I became very interested in this project. I was very happy we'd done this, and we'd had the good idea to follow this through. The other piece of information is that there are human knockouts of this gene. There are, in South Africa, there's a family described with albinism, and they had a loss of function in this gene. And in fact, it was even called albinos of dark skin. And I'm like, are you kidding me? This has got to be us, So we took the gene, we sequenced it, and again, we got very lucky. We found one and only one difference between our cases and controls, and it turned out to be an amino acid change. So if you remember, DNA encodes RNA, which encodes protein, and then that protein difference is what gives you the change in the cellular machinery. This is a type of change from arginine to cysteine at residue 93. And one way of getting a sense of how important this change is that you can do a sequence alignment. Here's that sequence alignment. I'll just read these out. This is human rhesus, which is monkey, mouse, dog, elephant, possum, chicken, out to zebrafish. You can see that all of these critters have an arginine at this position, and our kids are walking around with a cysteine, right? You don't need to do a very sophisticated mathematical model to know that this is likely not to happen by chance very often, and it's likely to be a big effect, okay? Biochemically, this is a very big change from one kind of amino acid type to another. We don't need to just rely on the models because, of course, we looked at 1,000 kids. So now what we can do is stratify by genotype, and the results are pretty beautiful. Here are kids that are homozygous arginine. Here are kids that are heterozygotes. And then here are kids that have homozygous cysteine. Here's the normalized skin pigmentation distribution, or hair pigmentation distribution. You can see that this homozygote and these heterozygotes are pretty much identical. So this is a very classical Mendelian trait where those that are homozygous are like 2 standard deviations out, right? You don't normally get this kind of stuff in genetics, and certainly not at this scale. Furthermore, the mutation is at 30% frequency in the Solomon Islands, and the frequency of the trait is like 10%. So it matches beautifully. If you remember Hardy-Weinberg, right? P squared, well, square 0.3, you get 0.09, which is exactly the incidence of the trait, okay? So we were very, very happy with this result. And what really kind of knocked it out of the ballpark is the fact that the only people in the world that carry this mutation are blonde Melanesians, okay? If you happen to be European, and this is what one might call chemically induced blonde hair, but if you happen to be natural blonde hair, you don't have a TERP1 mutation unless you're from Melanesia. You'll tend to have an OCA2 or a KIT ligand mutation. So here's blonde hair, all right? You don't have to be an MD, PhD dermatologist to study this trait. You have to be about 8 years old and look and see that people have different hair color and just ask a very simple question. And when we look at two populations, the two populations in the world that have blonde hair, Europeans and Melanesians, when we look at the second one, they've got blonde hair for a totally different genetic reason. So why would we suppose that diabetes is going to have the exact same genetic basis all over the world? Right? It makes no sense. We know there's going to be some things that are shared, but we also expect, especially for a trait like diabetes, where if you think about, again, India, South Indians get diabetes at a much lower body mass index, so at a lower sort of level of obesity than Europeans. And the same is true for the Japanese. And now that we've begun to study this trait in the Americas, we've found mutations that are private to Mexico, right? And so you say, Jesus, how do we not study this in each of these populations, especially for a trait like diabetes, which is like the #1, health concern for most economies going forward? Okay? Here is the other biological information. The TERP1 also causes this difference in pigmentation in this classical mouse model. In dogs, which we've studied, these dogs are also different for TERP1. And here's that family from South Africa that I mentioned where mom and dad are darkly pigmented, but the kids are albinos, because they have a knockout of this gene. All right, so all of this is really about beginning to think about all of the ways that we can bring external information to bear in trying to address these questions. So I want to sort of conclude this first part of the talk by saying that model systems are going to remain really key, even when we can sequence every human genome, even when all of you have your genome sequenced. Our ability to interpret it is totally based on the biology that we can understand. So there's a huge sort of, mashup problem in thinking about how to bring all these sources together, coming up with appropriate models for how to do that. To me, it's astounding that we do not yet understand the genetic basis of skin and hair pigmentation, one of the most highly heritable traits, one of the traits that we know is most important both evolutionarily and biomedically, right? Light skin pigmentation absolutely increases, your risk to skin cancer, and TERP1 is actually one of the genes that's most overexpressed in metastatic melanomas, right? So, really this is kind of important to understand for very fundamental reasons. And the cost of doing this is not very expensive. Each of these arrays, we just designed a new array for a company called Illumina, which we're selling, or Illumina is selling for $75, that looks at 2 million markers in the human genome. And we specifically designed it to be able to do trans and multi-ethnic mapping. We're going to use it on 50,000 people, but we're sort of offering it to the community so that people can use it in lots and lots of other populations. And so it's really about just sort of going out and asking these questions and having the kind of confidence that, if we go out where other people aren't going, we are going to be able to learn something totally new, right? And for me, it's been this sort of recurring theme of like, what are people doing? Okay, I'm going to do something at 90 degrees to what they're doing, because there's always an opportunity where other people aren't going. Okay, this now turns to DNA, to ancient DNA. I would say that ancient DNA is absolutely revolutionizing our ability to study the historical past, particularly of different human populations. You can't open up an issue of Nature or Science or even the New York Times without seeing one of these ancient genomes that's been sequenced. It really is an absolutely revolutionary technology. The fact that we could sequence a Neanderthal genome is just absolutely freaking amazing, right? And that we can put Neanderthals in the context of human genetic variation and variation from our primate relatives, to me, is astounding. One huge problem, though, is that almost all of the ancient DNA that's been done is from dry, cold climates, i.e. Europe, and that you need extraordinarily well-preserved specimens, or you need to be sequencing Neanderthals where the German government's like, I don't care if it costs $10 million, it's a Neanderthal, we're going to do the Neanderthal, right? If we want to think about broadening the set of samples we can study, it's a big problem because this is extraordinarily expensive, in large part because less than 1% of the DNA that you get from an ancient sample is human or human-like, which means it's literally 100 times more expensive to sequence an ancient genome as it is to sequence a modern genome. Right? If I take your blood cells, I get 99% human DNA. If I take a Neanderthal, it's like sequencing dirt with a little bit of human DNA in it. Okay? We don't have the German government backing us, and we're cheap, and we're also at Stanford where we like to be kind of disruptive, and so we said we got to figure out a way to come up with a new approach, and that's what I'm going to tell you about, and it's been an incredibly cool ride. We actually just spun out a company to do this too, and I want to kind of tell you all about it because it's what I wake up to every day and I'm most excited about. Okay, so before I tell you all that, I want to tell you a story about how we got into this game in the first place. How many of you have heard of Otzi the Iceman? Anyone heard of Otzi? Awesome. All right. So Otzi is an incredibly interesting case. Otzi is a frozen mummy who was found in 1991. The guy was frozen into a crevasse, into sheer ice, and two hikers came upon this guy, and they thought he was a hiker who died a couple years ago, right? So they start like unfreezing him and taking him out, and then they're like, wait, why does this hiker have a copper axe? Well, that's kind of weird. So they put that aside, and they're like, wait, he's got a bow and arrow and quivers. Huh, this may not be a hiker, right? And they look at it, turns out he was 5,000 years old. And he's been frozen in ice, literally like a frozen caveman, okay? And when they took him out, he was on the border of Austria and Italy, like 80 meters from the border. And so they wanted to figure out, okay, is he Austrian, is he Italian, is he Austrian? Because they wanted to set up a museum and and ride the Otzi wave. Okay, it's been awesome to study him because he's so well preserved. We know that Otzi's last meal was like ibex and einkorn wheat, so the guy was living like the paleo lifestyle with a little bit of wheat on the side. He died in his 40s, and we could do scanning tomography, and the guy had atherosclerosis, right? So, we all think, oh, the paleo diet, the paleo life. This dude really lived the paleo lifestyle, and even he had atherosclerosis. And what's cool is that we could get DNA from Otzi, sequence it, and show that he actually had a genetic predisposition to atherosclerosis, right? So it's all a little wild. As an aside, when we did this, we were going to send it to Nature. Nature sent it back, and we had colleagues who said, I've talked to the New England Journal of Medicine, and they're potentially interested in this. And they wrote it up in the weirdest case report you could imagine. A 5,700-year-old patient presents with, an arrowhead lodged in his back, and we sequenced its genome just to figure it out. It didn't get in the New England Journal; it went elsewhere. But anyway, so when we sequenced Otzi's genome, we wanted to place him in terms of European diversity. And several years ago, we'd had a big project on Europe where we had taken, and this is a principal component analysis of a giant matrix that's got about 500,000 columns. Each of those columns is a location of the human genome, and about 3,000 rows, where each of those rows is an individual. So you take that matrix, which has zeros, ones, and twos in it, depending on whether you match the human reference or not, and when you look at the principal components, it eerily looks like a map of Europe, right? So here, and these are all people that had 4 grandparents from a given country. So here's the Iberian Peninsula, here's the boot of Italy, the Swiss are nice and neutral in the middle, the Greeks and the Turks are near each other, which is good for us, not always good for the Greeks and the Turks. The US and the UK even cluster in our special relationship. So when we think about Otzi, we would expect him to sort of PCA out here, right? He's sort of from northern Italy. Turns out he PCA'd out here with these Italians that we'd always kind of wondered about. We were always like, what's up with those Italians and why the hell are they over here? Turns out they weren't just any Italians. They were Italians from an island called Sardinia. And Sardinia we believe to be a remnant population. And furthermore, the real kind of key was that when we looked at Otzi's Y chromosome, he carried a mutation that is only seen today in Corsica and Sardinia. So here's a 5,000-year-old dead guy, and because we've studied Europe so much, I can literally find a town in Sardinia with his closest living relatives, right? I can't do that for most of you. And so I've gone out and said, we need to have something called the Otzi rule, right? Whatever we can do for good old Otzi, we should be able to do for other people alive today, and that's what we're working towards. So in order to kind of move this forward, this postdoc named Meredith Carpenter, this absolutely brilliant postdoc in my lab, came up with a strategy for enriching human DNA, okay? And it's really quite an amazing technology because it allows us to take these ancient DNA samples and almost as if we had a magnet, just suck up the fragments that match human DNA. This was done in collaboration with a PhD student, Jason Buenrostro, who happens to be Latino, and Will Greenleaf, who probably is not Latino, who is my colleague at Stanford. The 4 of us really sort of came up with this. We've actually filed a patent on it. We filed several patents on it. The technology's now been licensed by a company that I'm one of the founders of called Identify Genomics, and it's been a really super fun ride. So if any of you are interested in starting companies, I can tell you about that too. So we did this technology and then applied it to really the first big paper on more than one ancient genome. So we had 4 teeth from Bulgaria, bone from Peruvian mummies, and a hair sample from Denmark. And then we intersected it with these data from 1,000 genomes that I was telling you about. And I just kind of want to walk you very quickly through the way the technology works. So for those of you who know a little bit about biology, you know that one of the things that we want to do is take DNA and try to match DNA. And there are 2 ways to do that. We can match with DNA or we can match with RNA. And we chose to match with RNA for the reason that first, RNA DNA hybridization is a stronger hybridization. And secondly, we're trying to study ancient human DNA. The last thing we want to do is put in modern human DNA because it's going to be a contaminant. Okay? So we came up with a technique to make boatloads of human RNA that come from the human genome. And this involves taking a DNA library, adding something called a T7 promoter, which then we can just transcribe and transcribe and transcribe and make boatloads of RNA. So the way this normally works is you take an ancient tooth, you do a DNA extraction, about 99% of it are these black fragments, which are environmental DNA, bacterial DNA, 1% is this human fragments, and then we can make, these are called sequencing libraries, that's what goes on the sequencing machines. So we take these sequencing libraries and we hybridize with our RNA, and these things have something called streptavidin beads that can capture this thing called biotin, and then we can fish it out. We use a magnet to fish these things out. Everyone kind of have a sense of sort of how this is working? All right. So here are the applications. As I said, we had 4 teeth from Bulgaria, where we're now studying about 200 different samples. Here's a Peruvian mummy who's horrified. There has yet to be an equation in this entire talk. And then here's our, I don't even know if there is an equation in this talk. And then here's our sample from Denmark. It's literally this Viking-looking guy. Okay. Here are, it almost looks like, advertisement. Here are the proportion of reads that map to the human genome, pre and post capture. So in blue here, you see that on average, about 1% of it maps to the human genome, which is what you would expect. This is variation from library to library. So these are all the Native American samples, and these are some of our Bulgarian samples. And then post capture, you can see how much better we're doing. Right? So we're not getting 100% human DNA, but we're doing boatloads better than we were before. In fact, the amount of enrichment, and again, this is like almost out of that book, How to Lie with Statistics, is about 140-fold, right? So you can choose different ways of measuring this. For some of these, we've gone up 140-fold on the amount of DNA that we're getting. It's actually, so one of the things that is important is that it's pretty randomly distributed. So in blue here is what you get pre-capture, in red is what you get post-capture. There are different ways of quantifying this. I don't sort of want to get into the details, I just wanted to give you that kind of visual. And then one of the things that's actually very interesting, and for those of you who are looking for a good problem, is to figure out, and this actually relates all to the issues of reproducibility. The reproducibility in science is a huge hot topic right now. And so here's an example of where we've sequenced the library pre-capture and post-capture, and about 80% of the fragments that we find in the pre-capture library we find in the post-capture library. But of course, this is actually very much related to what is classically the capture-recapture problem, right? So we've gone out fishing once, and then we went fishing the second time. These you can think of as the marked fish or the marked rabbits or whatever, and then here are the ones that we get, the ones that are marked and the ones that aren't marked. And of course, your expectations about the overlap depend on, well, how much did you fish here relative to how much you fished here. Does that make sense? Okay. And in fact, these curves, this is what you get. So it's related to this problem of, here's the amount of sequencing I can do. I have a library which has a finite number of molecules in it. Right? The more I sequence, obviously, the more likely I am to see a duplicate, right? Because I may just fish out the same molecule by chance, right? So here's the number of unique fragments, which we expect, of course, asymptote, right? If we sequence in the limit, we'll get every molecule that's in there, and we'll have seen every molecule at least once, okay? So that's what happens pre-capture. We can think about this technology as basically moving you up on this curve. So, for the same amount of investment in sequencing, instead of being here, you're sort of somewhere out here, okay? And this is the ratio of those, which is the fold enrichment. And of course, it just stands to reason that the more I sequence, the less enrichment I'm going to have, right? Because it's, eventually these two curves are going to meet, right? And it's the ratio of the two curves. So I can tell you more about that model, and many of you probably have better ideas on how to do this, but there are lots of kind of interesting problems that relate to this. Okay, another way of thinking about this is, because what we care about are so-called SNPs, single nucleotide polymorphisms or variants, is for a given amount of investment in sequencing, say 1 million reads, how many SNPs do I find? Pre and post capture. And the point of this is that we always do better with the capture. at least 2 to 3-fold better, in some cases 14-fold better. In terms of the metric we really care about, which is the number of usable variants in the human genome that I can then intersect and learn something about ancestry with. Okay? So here is some analysis of these data. This is another principal component analysis. These, okay, I don't know why that's happening there. So these samples here in red come from Africa. These samples up here in green come from Europe. These samples out here come from Asia and from the Americas. And so what you're really seeing here is sort of the African corner of diversity space, the European corner of diversity space. This is us, Latinos. We're admixed, and so there's some of us that have more European ancestry, some of us have more Native American ancestry, some of us have more African ancestry, and these are the samples that we ourselves sort of collected. And your resolution on this depends on the number of markers. So here we've got 100, we've got 1,000 markers. We don't get that awesome a resolution. Here's our ancient sample, and what we can say is that this tooth from Bulgaria looks European, which is like, oh shit, okay, if it didn't look European, we'd be in a lot of trouble, all right? Post-capture, we've got about 10,000 SNPs, and you can just see how nicely these begin to tighten up, okay? And you could actually begin to place this within the map of Europe. Here's another example. Here's that horrified mathematical mummy that didn't get the equations. It actually turns out to be a super interesting mummy. This is a chichamoya, which are the mummies that are thought to have blonde hair, ironically enough. And some people have argued that they're likely to be of European or East Asian ancestry, which we don't totally believe. So we're now sequencing mummies all over Peru to try to figure this out. When you look at this mummy in this 1,500 SNP panel, which are the random set of SNPs we got in that sample, you actually see that it begins to cluster with the East Asians, maybe giving some credence to that crazy hypothesis. But when you go up post-capture, and again, this is the same amount of sequencing money, right? So you would much rather have the post-capture data. You see that it actually clusters very closely with the Aymara and Quechua, who are the indigenous group in Peru that you would expect them to cluster with. So it's actually pretty cool. And for those of you who are interested in those problems, we've got a ton more that we're working on there. Okay, so in summary, we've developed this new technology. It allows us to enrich DNA, in some cases going from about 1% DNA up to 60% DNA. We're getting about 50,000 markers per million reads. A million reads today probably costs you, I don't know, maybe $100, right? So it's actually a quite scalable technology to use. And we're capturing the majority of the fragments that are in the pre and post-capture library. Now that we've spun this into the company, we've actually improved the technology, and it's doing far better, and I can tell you about that. But in the last few minutes that I have, I actually want to tell you about, for me, some of the most important applications of this. And that has to do with the transatlantic slave trade. So the transatlantic slave trade is one of the most horrendous periods in human history. It is the forcible migration of over 12 million people from all over Africa. These people were brought to the Americas, as you know, as part of the triangular trade. The majority of them went into the Caribbean and Brazil. Only about 200,000 came directly into the port of Charleston. If you want to understand the history of African descent populations, you have to study it first in the Caribbean and Brazil, and really understand the entire context, because it's truly sort of horrifying. African Americans are looking to DNA as one of the sources to help them reclaim part of that history. Some of you may have seen Skip Gates' show on tracing your roots. I've worked with him as part of that show. We actually did Jessica Alba's dad's genome as part of that project, which is really interesting, as well as other individuals of admixed ancestry. And in particular, because we've lost so much of the history, they'd like to be able to take DNA and match in Africa what populations an individual today is most closely related to. Turns out to be a very difficult problem, because if I think, for example, Oprah Winfrey has done this, and they tell Oprah she's Zulu. Well, I can tell you there's no way Oprah Winfrey is Zulu, in the sense that she's got many ancestors that contributed to her, and the odds that all of them were Zulu is, epsilon, where you let epsilon get very small, right? And so, maybe her mitochondrial genome is most closely related to the Zulu tribe, but that doesn't mean she's Zulu, okay? And so in order to answer this question, and we've studied this to death, we've studied lots and lots of African Americans, and what we can basically tell them is you're about 80% West African, 20% European, or whatever the numbers are, that's the mean, but it's very, very hard to say, okay, this is the amount of Yoruba you have, this is the amount of Igbo, this is the amount of Hausa, and that kind of thing. Enter now these set of samples. So in 2010, there's a project in the island of St. Martin's where they dug up a slave cemetery. And in that slave cemetery, there were a set of skulls that we got access to as part of a collaboration. Here's one of these skulls. You can see that these skulls have a very characteristic dental modification pattern. This is a dental modification that you see in African tribal groups. You don't see this in American populations after the slave trade because their culture gets quelched and killed, right? So they're not keeping their tribal practices. The fact that this individual has these filed teeth tells you that they likely were born in Africa and they were part of the Middle Passage. This person was on a slave ship and came to the Americas. Furthermore, we can use isotopes to demonstrate that this individual lived most of their life in Africa. Okay, so this is just an amazing potential route into understanding the slave trade. Problem. Most of what we know about ancient DNA is based on cold, dry samples. That's where DNA is most preserved. St. Martin's is hot and wet, right? There shouldn't be DNA in there. And in fact, there's not a lot of DNA in there. And that's why we needed to use our technology to fish out as much DNA as we could. When we looked, there's one sample. The woman leading this project is Maria Avila, and she looked at 3 of these samples in collaboration, Hanne Schroeder in Denmark. And this individual in particular is a male. When we looked at his Y chromosome, we initially got a little concerned, because his Y chromosome is of a group called R, for those of you who are aficionados of Y chromosomes. This is the distribution of the Y chromosome. Even if you're not an expert geographer, you'd know that this is not Africa. And so you'd say, okay, you guys, you got some contamination in there. It turns out that he carried a rare haplogroup of R1b, which carried a mutation that today is only seen in the Sahel. It's only seen in Cameroon, very similar to Otzi, okay? So here is the first kind of breaking down of that Otzi rule. We were able to take this individual of African ancestry, first get usable DNA, and then secondly, map his at least male lineage to a very narrow geographic place. Because we had his whole genome, we could also play our PCA games. Here's that sample. In fact, we can make a tree of all the Y chromosomes. It clusters with a set of samples, ironically also from Sardinia, that were probably part of the Roman slave trade. And these guys coalesce about 10,000 years ago, or 8,500 years ago, which makes tremendous sense in terms of the history. We can now take STM1 and map them back to Africa, and here are the set of populations that we sampled. I don't expect you to worry too much about the labels, but what we could show is that STM1 clustered with the Bamoun, who are a tribal group that today lives in Cameroon. Okay, so again, this is really quite incredibly high resolution, given that it's an African sample and given that it's ancient DNA. This paper was recently published in PNAS about, I don't know, a month ago. Okay, I want to end now on our latest adventure. Lots of people study the transatlantic slave trade. There's a lot of history to the transatlantic slave trade that we can work off of, including the digitized slave ship records. I have a postdoc, Rosie Frehel, who came to the lab very interested in the Indian ocean slave trade. And I was like, I didn't even know there was that slave trade, right? And I'm supposed to know this kind of stuff, right? It turns out that during historical times, the Indian slave trade also moved nearly 1 million people. And in particular, she works in this very remote part. And islands are awesome in biology, as you know. Islands are always very good model systems. And she works in Mauritius, which is a tiny little island off the coast of Madagascar. And what's interesting about Mauritius is that it was not inhabited until colonial times. Okay, so the French take this over, and being the French, they got to bring in the help, and so they bring him in from Madagascar, they bring him in from East Africa, and that goes on for about 100 years. Okay, so here's Mauritius. This is Krish Sita, who's one of my colleagues at Stanford. Krish is in the anthropology department. This is Rosie. And so the slave trade lasted for about 100 years. It included people from Madagascar, East Africa, and West Africa. After the slave trade was ended, they then brought in indentured servants, also kind of like slaves, brought in from South Asia, East Asia, and Southeast Asia. And you then had the French, and the French were going to want to contribute genes too. And so you really have this very interesting admixture process that's happening both in the context of the slave trade and one in which we'd like to be able to understand this kind of complex coming together. Mauritius is of particular interest to us biomedically because it's got one of the highest incidence of diabetes in the world, something like 50%. Okay, so what have we learned? Well, we looked at 2 sites. The first site is really amazing and horrific. It's a site called the Lamorne Peninsula. So here's Mauritius. The Lamorne Peninsula juts out here towards the end. It's incredibly staggering, 2,000 feet above sea level. And during the slave period, the slaves would hide in the caves up in the Lamorne Peninsula. After they were freed, the police came over to tell them they were freed. And they were so scared that they jumped to their death. So, we were able to get access to this slave cemetery at the bottom. They then interred other individuals there. And then there's another cemetery where they buried the indentured servants, so we've got samples from both. And this has been a really quite amazing project. Here, this is still unpublished work. Here are the samples from the Lamorne Peninsula. Pre-capture, again, less than 1%. Post-capture, one of these samples had like 45% human DNA. Another one had 12%. The Bois Marchand site, which is the site for the indentured servants, is not nearly as good, but we were still able to get some. The mitochondrial haplogroups, so the mitochondria come from your mom, for all of these from the slave cemetery were of sub-Saharan African ancestry. Totally consistent with them being African slaves from West Africa. But then the autosome, which has both mom and dad's ancestry, shows you that complex history, and I'll show you a PCA in a moment. Whereas the Bois Marchand site that had all the indentured servants, all of their mitochondria were from India and Southeast Asia. Again, totally consistent, right? So first we can get DNA out of the tropical samples, and they make sense, and they really are going to help us understand these complex histories. So this is, I think, close to the last slide. So here's, again, our principal component analysis. Here are our West African samples. Here are our East Asian samples. Southeast Asian, East Asian, Southeast Asia. Here are the Indian samples. And then the Lamorne Peninsula, again, were the slaves. Here's an individual that you can see is like a first-generation admixture between India and the Southeast Africa. You're seeing this, the first-generation admixture event. Whereas others are pretty much West African, Southeast African. The Bois Marchand site, which is where the indentured servants are, here these guys are clearly, Indian Asian ancestry. And this is another individual who is basically, a cousin to this guy, at least genetically, right? So we're beginning to peel back the onion, all the layers of admixture that happen. So we're very excited about this technology. We are trying to now get samples to other slave cemeteries in the Americas. You can imagine beginning to study the antebellum South, and you begin to see, okay, when did this admixture event happen? How much of it happened during the antebellum period, which we believe is one of the biggest contributors. So the fact that a quarter of the DNA in an African American individual, so basically a grandparent, is of European ancestry, we believe that really dates back to the control that European men had over the plantation, and just how much of that really happened all the time, the sort of Thomas Jefferson effect, if you will. And it was, more the norm than the exception, right? And it's just kind of horrific to think about. And if you think about sex bias patterns most of the Y chromosome, for example, that those of us who are Latinos carry tend to be European. Most of our mitochondria tends to be Indian, Native American, or African, right? And so it's telling you exactly how that process occurred. All right, with that, I'd like to conclude and tell you it's really possible to get genome-wide DNA from the tropics and reclaim detailed genetic picture of the slave trade. We're getting closer and closer to realizing the Otzi rule, and nothing would make me happier than, we're now seeing, after we've clamored and others about diversity in medical genetics, now, Obama in his precision medicine initiative has made diversity one of the keystones, and it's one thing I'm always harping on NIH about. I think this is also going to spread into other areas of research. It looks like STM1 was a Bantu speaker from Cameroon. I didn't tell you about these other two, but they seem to be non-Bantu speakers from a different part, so they're all buried in the same cemetery. It gives you a sense of just how these people were treated, and that also our work now on the Indian Ocean slave trade is yielding very interesting insights into this sort of three-way admixture that also included individuals from South Asia. I'd like to end by acknowledging the folks who did all the work. Meredith developed the technology along with Jason and Will. Maria has done all the analysis on the STM1 samples and the slave trade stuff. Dave Posnick is an incredible guy in my lab who does all the Y chromosome work. Alex is my lab manager. All this work's done with a lot of the ancient DNA work in collaboration with this crazy Dane named S.K. Villershev. He's really sort of a force of nature. We share this postdoc named Morton, who's sort of our bromance child there, and he helps us do all this work. All right, I'm happy to take any questions you might have.